So potential energy is stored energy. We find this in our gasoline. We find this in the biological molecules that we eat on a day-to-day basis. Pretty much any time you have atoms sharing electrons with one another, you have potential energy. All right, so what's kinetic energy? Well, kinetic energy is when it's being used. So the use of potential energy, or the manifestation of energy, or the movement, as we've talked about, the actual work is being done. So when we see any type of movement whatsoever, that's kinetic energy, whether it's movements of tiny atoms or whether it's movement of a large conglomeration of atoms like you and I. So any type of movement in our universe is kinetic energy. The ability to create that movement but not being used, that's potential energy. Now there's other types of particles that exist in nature. Heat, light, electricity. This deals with different manifestations of subatomic particles within atoms, or the actual electrons themselves. So light is photons of energy. They're pure energy that get emitted by the sun and other types of reactions that can release that type of energy. Heat is also radiant energy, where molecules can dissipate those types of particles, and they can be absorbed and sometimes be reused. But that's really what kinetic energy is. It's just movement of any and all types of particles and waveforms and whatnot. So that's it. It's either potential or kinetic. If it's stored, it's potential. If it's being used, it's kinetic. And you can convert one back into the other. So here we have a bicyclist who probably ate a good breakfast in the morning. So he ate a lot of molecules that had a lot of potential energy. Well, now he burns that by undergoing metabolism. He breaks the covalent bonds of that oatmeal or whatever he ate, and that provides the kinetic energy for him to go up the hill. Well, now that he's at the top of the hill, he has potential energy primarily due to gravity. And then that potential energy can be converted back into kinetic energy as he goes down the hill. So that's really all it is, whether it's bicycling or whether it's biological or whatever the case may be, it's one or the other. So it's always being converted into kinetic, back to potential, back to kinetic, and so on and so forth. Well, we know many different sources of energy. We've got batteries that have potential energy. We've got gasoline, which has a wide variety of potential energy. And we have basic biological molecules. And we use them in a variety of ways to fuel what we need. And you turn the light on, it starts using that potential energy and turns it into photons of light and such. The car can use it for pretty much everything we've talked about. Movement, heat, light, electricity are all generated by the combustion of this fuel. And then, of course, us, as we eat glucose and lipids and proteins, our mitochondria break these down into ATP, which is our source of potential energy that fuels our muscles and our neurons and the like. Now, let's talk about thermodynamics, because these are the laws that govern energy in our universe. Now, remember we talked about laws. These are a progression of the scientific method that we know so much about, we can make predictions on what's going to happen. So these are very well understood concepts that we know quite a bit about and can make these predictions based upon these laws. The first law is pretty easy. Our universe is essentially a closed system where we have no input nor exit of energy. So the first law is where we call it the law of energy conservation. Energy cannot be created nor destroyed. It doesn't come into existence, nor does it go out of existence, as far as we can tell. There are still unknowns about dark matter and dark energy and whatnot. And once we figure those out, we might have to adjust and modify a few things. But in all of the observable universe, as far as how things work, energy doesn't pop into existence, nor does it pop out of existence. It's always there. So only you can change it from one form to another. I mean, if you take a block of wood and you burn it, you may think you've destroyed the energy. You haven't. You've merely converted it into another form of energy, namely heat, light, and whatnot. So you've broken the covalent bonds, but you haven't destroyed the energy. You've merely transformed it. So that's the first law of thermodynamics. Energy is conserved, always. It always remains constant. Now, there's a second law that's a little more difficult. This is the one that we're going to focus on for biology, because it plays a key role in how the cells and other systems maintain homeostasis. Even though all energy is conserved in our universe, when you transfer energy from one molecule to the next, from one system to the next, it is always inefficient. Now, some systems, some energy transfers are more efficient than others, but there's always going to be that inefficiency going on. So what do we call that? There's actually one word that could be used to describe the second law of thermodynamics. I mentioned it before. Do you remember what it is? It's up there too. Entropy. So entropy is the second law of thermodynamics. It basically states that when you transfer energy from potential to kinetic, or kinetic back to potential, or potential back to kinetic, and so on, you always lose some to the environment. Now, some things are more efficient and therefore lose less, but that's always in play. Entropy causes things to become more random, is another way of putting it. My daughter's room never stays like this. It's much easier to have a high degree of disorder than it is to have a high degree of order. In order to get this, you've got to put energy into it. This is what occurs naturally. But on a scale of what is going on in our universe, there's another way of describing this. When we say things have a high degree of order, we mean that they don't, one way of describing that is saying that they're very highly concentrated. Let's look at an easy example. Let's say you have a perfume bottle. And as long as it stays closed, the perfume will remain in there for long periods of time. But as soon as I open up the top, what happens? You get evaporation into this outside environment. And so what happens is when you allow for the molecules to escape, then this is the natural order of things. Things tend to become less organized and more random. Another way of describing that is things go from a high concentration to a low concentration. The perfume is at a very low concentration out here in the atmosphere. And so therefore, its natural tendency is to evaporate and go into this very low concentration. Okay? Think about a canister of carbon dioxide, maybe like we have in a variety of different places. You don't naturally get carbon dioxide coalescing from the atmosphere and compressing into this vial. That's not what happens. But let's say you have a high pressure system of carbon dioxide here, and then you open it up, what happens? It'll escape to where you have lower amounts of carbon dioxide. Okay? So the same thing is true in biology. When we have molecules like ions that are unevenly distributed on one side of the membrane or another, then they tend to flow from a high concentration to a low concentration until they're equally distributed. Once they're equally distributed, then the net flow is zero. And that's really what entropy is all about as it applies to biology, is that if you have different concentrations of things, then they will try to equalize. That's really what it comes down to, what entropy comes down to. Okay, so in chemistry, we learned that if you have certain reactants but no products, then the natural tendency is that two things will react with one another to start generating some product. Well, as the product increases and as these decrease, then this essentially equalizes. So it's the same process in metabolism in our bodies. If we have substrates, which we call them, or things to interact and react with one another, and we don't have any of the products, this will naturally occur to create these products. But once we have the same amount of products as we do as the reactants, then the equation stops. Now, most of the time, we get rid of the product so that this keeps flowing this way. We go from a high to a low concentration. So let's show how this works. And this is how kinetic and potential energy work in a biological system. Some reactions absorb energy, some reactions release energy. So let's look at the absorption. When a reaction absorbs energy, it is said to go from kinetic to potential. And this is exactly what photosynthesis is. Light, which is kinetic energy, in chloroplasts is converted into covalent bond energy by covalently bonding carbons and oxygens and hydrogens together to form glucose. So this is taking the kinetic energy, which is just constantly flowing to our planet from the sun, and turning it into potential energy. That's really what plants do. This we have a special name for. We call it an endergonic reaction. So an endergonic reaction is a reaction that absorbs energy and is turned into potential. So it's kinetic to potential. This is how we build our structures. Your body has a substantial amount of potential energy that is built up over your lifetime. Okay? You have a lot of energy in all of your molecules. Exergonic reactions are reactions that release energy. For example, in your mitochondria, when your cells absorb the sugars and the fats and the proteins, the mitochondria will break those covalent bonds and release the energy that was stored in the glucose, in the lipids, in the proteins. So this is the equivalent of going from potential to kinetic. An exergonic reaction is breaking the bonds, releasing energy by converting it into kinetic energy. So you can see how these two reactions essentially work hand in hand. Some reaction releases energy, an exergonic. Another reaction absorbs that energy, endergonic. When we put those two together, let me jump ahead real quick, we actually call it a coupled reaction. So what is a coupled reaction? A coupled reaction is nothing more than one reaction releasing energy, exergonic, and another reaction absorbing that energy, endergonic. That's it. So here's an example. ATP, that battery that I told you about, and I'll show you how ATP works in just a second. ATP provides the energy by, when its covalent bond is broken, it gives the energy necessary for actin and myosin, which is in your muscle, to interact with one another. That's what causes muscle tension. So when ATP uses its energy, then the muscles absorb that energy. The muscle contraction is the endergonic reaction. When you break ATP, that's the exergonic reaction. And so, something as simple as flexing your muscle is a coupled reaction. Something gives up energy, the other absorbs it. All right? I might add and point out how entropy plays here. Heat is mostly the byproduct of inefficiency of energy transfer. Remember I told you that entropy, when you transfer energy from one system to the next, there's always going to be some loss of energy. So the muscle doesn't fully claim all of the energy from the ATP. Some of it dissipates to the environment as heat, which is why when you exercise, your body heats up. Because every time you break covalent bonds, you generate heat as a byproduct due to entropy. All right, so let's look at why ATP. Why does the cell use ATP instead of just directly using glucose or lipids or proteins or whatnot? Well, remember what we talked about with covalent bonds. The reason why atoms form covalent bonds is because it provides stability. Valence electron shell theory. When the atoms have a full valence electron shell, then the atoms are stable. So in order to break a covalent bond, you have to overcome the resistance that these atoms have to becoming unstable. They don't like that. They like to hold on to those electrons. They like to stay close together and share the electrons to form those covalent bonds. That's why it's a good source of potential energy. Well, glucose, fats, proteins, they're all the same. They're very stable molecules when it comes to the covalent bonds. So your mitochondria are the organelle that are specifically designed to take any organic molecule and turn it into this. So why does this become the universal battery that the cells need to be able to transfer energy? Well, it comes down to this portion right here, the triphosphate portion of the ATP. Or remember, it stands for adenosine triphosphate. So why is that so important? Why is this so special? Because energy can be found anywhere. You got energy here, there, everywhere. Why this? Well, look at these phosphates. What do you notice about them? What do they have? What do they have in the top part of this? What does this little negative sign mean? They're charged. Okay. What happens when you put molecules of the same charge near one another? Do they attract or repel? They repel one another. It's not like an ionic bond where you have two opposite charge ions. You've got three ionic charges here that are very strong, repelling one another. Well, due to that fact, the covalent bonds that hold these phosphates together become weakened, and as such, become the easiest covalent bonds to break in any biological molecule. And that's why your cell converts all organic molecules into ATP. Because the cell, the machinery of the cell, can tap into this energy anywhere. Okay? It doesn't need any special type of thing. This is easy energy, the easiest. This also makes the ATP molecule very unstable. So your cell usually doesn't make ATP except for in the moment when it needs it. So your mitochondria are just constantly pumping out ATP, and your cell is constantly using that, especially your muscles and your neurons. Okay? So that's why, and that's one of the questions I'll ask you. Why does the cell use ATP instead of directly using glucose and other sugars for their covalent bond energy? It's because of the nature of these three phosphates. They create this repulsion field that makes it so that that covalent bond is very easy to break. And the trick is it has just as much energy as any other covalent bond. It's just easier to break, and therefore easier to extract the energy from it. So guess what? ATP acts as an intermediary between most reactions. For example, you eat sugars and fats and proteins. First, your mitochondria is going to break them down. That's an exergonic reaction. And assemble used up fuel molecules, which we call ADP, adenosine diphosphate, which has two phosphates instead of three. And it just reattaches that phosphate. So this is an endergonic reaction. Remember what happens when we put the two together? We call it a what? A coupled reaction. So you release energy and it gets absorbed. Now this becomes the exergonic reaction. Now the ATP releases the energy somewhere in the cell and something else absorbs it. And that becomes the coupled reaction. So you can see that all it is just energy transfer. It's just going from one molecule to the next to the next. Okay? As I just explained it. Your cells absorb glucose. Your mitochondria will break the covalent bonds of that glucose molecule into its fundamental molecules, carbon dioxide and water. We'll learn that this is exactly what plants need to make glucose. So when the mitochondria break the glucose back down, they turn it back into the fundamental molecules that the plant needs to, again, remake it into glucose. So the cycle just keeps going round and round and round. We consume glucose, plants make it. We consume it, they make it. Notice, though, however, that as we transfer energy from glucose to ATP, and from ATP to, say, making a protein, which requires energy to assemble the amino acids together to form a polypeptide chain, notice energy is lost. What's that called again? What law? Entropy. That's entropy right there. You can never fully transfer all of the energy from one molecule to the next. There's always going to be some loss of energy. Which brings us to the fact that if we don't have photosynthesizers on this planet, you and I wouldn't be here. Because we'd run out of energy. Without something to absorb the sunlight and turn it into food for you and I, we're not here. We don't exist. We cannot survive because we don't have the capacity to generate sugars and fats and proteins from light. Plants and other photosynthetic organisms do, hence our dependence upon them for our food. Because you can't recycle all the energy in your body. If you did, you wouldn't have to eat hardly any, at all, if you could recycle all the energy without any loss. All right. ATP is very, very short-lived, okay? But the cell can pump out mass amounts of it for your glucose. I mean, it can take a single glucose molecule and almost pump out 40 of these, okay? So you get a lot. You get a lot of ATP for your organic molecules. Okay. Now, the last concept today, enzymes. And we probably won't get all the way through this, but I've got some videos to show you, as well as some things we did here at UVU that really help out with this thing. Now, when we talked about dehydration, synthesis, and hydrolysis, really we were talking about these two processes right here. For example, when you break covalent bonds and split polymers down into monomers, that's called what? Is it dehydration, synthesis, or hydrolysis? Hydrolysis. Guess what? Hydrolysis and an exergonic reaction are the same. They're exactly the same. That's all hydrolysis is. Hydrolysis is the breaking of covalent bonds and the releasing of energy. So guess what dehydration synthesis is? It's an endergonic reaction. When you covalently bond monomers together to form polymers, you are building complex molecules and storing energy as potential energy. So now you know that dehydration synthesis and hydrolysis are equivalent to an endergonic and an exergonic reaction. That's really all it is. But these two processes in biological systems don't occur naturally. And the reason for that is because, remember, covalent bonds are stable bonds. In order to break one, you have to do one of two things. You either have to provide enough energy, usually by heating something up, to cause them to break apart. You know, they absorb that energy and eventually they'll break apart. Or you just lower the requirement for the atoms to break that covalent bond. Well, guess what? We have biological structures that do the latter. They're called enzymes. So what do enzymes do? Enzymes are proteins, which are essentially, as we learned in the organic molecule lecture, polypeptide chains of amino acids that fold into a tertiary, or sometimes quaternary, depending upon how many there are, structure. And based upon that structure, they each have a certain job. Some enzymes undergo dehydration synthesis, where they assemble molecules together. We've gone over a major enzyme in the last lecture. Anybody think about it? It's an enzyme that actually makes enzymes. It's ribosomes. Oh, I thought you said ribosomes. You said lysosomes. Ribosomes. Sorry, I'm jumping the gun. Ribosomes are enzymes, because what do? They assemble amino acids together to form polypeptide chains. So they're literally enzymes that make other proteins. Which brings us to a good question. Which came first, the enzyme or the protein? You know. So, and that's a good question. We've asked that one, and there's an answer, but it's not for this class. Well, later. We'll get into that. So, some enzymes have a particular structure that undergo hydrolysis. For example, in your mouth, you have an enzyme called amylase. And when you start eating foods that are especially high in starch or carbohydrates, that amylase starts breaking down the starches into disaccharides. And then once they enter in your stomach, there are other enzymes that break the disaccharides down into monosaccharides. And then when it gets into the mitochondria, there are enzymes there that break down the monosaccharides and turn them into ATP. So for every stage of breaking covalent bonds, there's an enzyme. Okay? Now, what do enzymes actually do? Well, think about it. Imagine that you were trying to assemble your kid's bike for Christmas. I didn't buy my kid's bike for my kids this Christmas, so I didn't have to assemble anything together. But imagine back in the day when you had to assemble them. Now they come prefabricated. Let's say that you had to tighten the nuts and the bolts and whatnot. and you didn't have any tools whatsoever. Could you do it safely, where your kid wouldn't fall apart on his bike? Probably not. Why? Because without the tools like a wrench or screwdriver or whatever, you wouldn't be able to facilitate the necessary torque to tighten the nuts, to tighten the bolts and whatnot. Well, the same thing is true for enzymes. If you don't have an enzyme, then the job that enzyme was supposed to do really can't be done in your body. Because in order to get it done without an enzyme, you have to heat your body up so much that what starts happening when you heat proteins up too much? They denature. So you can accomplish the same thing by generating lots of energy, but it would normally kill you to do so. So what happens is, when you either break a molecule apart or put molecules together, there's an inherent amount of energy required to accomplish that. And this is a key term that you're going to have to know. It's called energy of activation. It's not a new type of energy. It's just the amount of energy required to break or make a covalent bond. That's what activation energy is, or energy of activation. The amount of energy required to break or make a covalent bond. Now, I have a very simple example that illustrates this concept. Let's say that you want to study for one of my quizzes with someone here in the class, and they've been very reliable in the past, and you really need their help. Well, one day you say, hey, let's meet at the top of the hill and study and do what we need to do. Well, you get to the top of the hill, and you notice that they are, they found some new person to hang out with, okay? So they decide to go off and leave you alone and to fail my quiz. Well, there just happens to be a boulder up here, conveniently, in this position, and you decide to push that and ultimately end your misery by ending theirs. Now, that's a hard thing to do, to push that large boulder. I'd rather get my revenge an easier way. How can I do this easier? What should I do? What is that? Leverage, fulcrum, a lever. That makes it really easy to get that ball rolling and kill them. So, in the end, what is this? That's the enzyme. The enzyme allows you to do the same job with less energy input. It's like trying to screw in a nut with your fingers versus a wrench. One's substantially easier than the other. Okay? Requires substantially less energy to get the same job done. That's what enzymes are. You can think of it as the energy required, say, to push a boulder down a hill or to get the ball rolling. Once you get the ball rolling, then the energy releases itself and you get more out of it than what you put into it. But you still have to overcome that initial resistance of these molecules to break their covalent bonds and whatnot. So that's where enzymes come in, because at normal biological temperatures, the amount of energy required to spontaneously break covalent bonds is not sufficient. As such, enzymes are proteins that have a particular tertiary structure that lower the amount of energy required to break the covalent bond. But they are specific to what we call the substrate, which is what they metabolize, what they break down, what they put together. This is not just breaking things down, it's also putting things together. The same principle applies. So that's what a catalyst does. It essentially lowers the amount of energy required to cause some reaction to occur. So it's like I mentioned before with the wrench and the bolt and the screwdriver and the screw. The wrench can't do the screwdriver's job and vice versa. And if you try to do it without either tool, the amount of energy required is substantially more than if you have the proper tool to give the necessary torque and other things of that sort. This was what I explained is how you know that if you have too much heat, too much change in a pH, and too much salt or a different salt concentration, that ultimately denatures the enzyme. The enzyme cannot function, as you saw many examples of the liver in that. Now, we're going to describe and explain why cyanide kills you, as well as many other various toxins or whatnot. Because the things that are toxic to you and I aren't necessarily toxic to every living organism, and vice versa. Antibiotics. Why do antibiotics hurt bacteria and not us, barring an allergic reaction, which I'll explain the difference here in a second. So, enzymes, in order for them to do their job, that tertiary structure must be unbound to something else. It has to be free to be able to break something apart or put something back together. So there are some poisons for any organism that essentially mimic the substrate that the enzyme is supposed to metabolize, but because they're not quite the same structure, then they end up sitting where the enzyme is supposed to do its job, and they are not metabolized, so they just sit there, and they prevent the metabolism of that particular product, and the enzyme needs to literally be destroyed and remade before it can function again, which takes a little while. So why does cyanide kill you? Well, there's an enzyme in your mitochondria called ATP synthase. Guess what it makes? ATP. Cyanide blocks that enzyme from being able to make ATP. And so when you get sufficient amounts into your bloodstream and into your cells stop producing ATP and you die. So that's why cyanide kills you, as well as many other substances can actually do the same thing to another enzyme that's critical for your cell survival and energy production and things of that sort. Let's look at penicillin. This was actually a secretion from a fungus that fungi used to kill bacteria and cause them to die and then absorb them. So this is how we first discovered antibiotics, is actually fungal secretions that they use for decomposition. Penicillin was one of these. So why does penicillin kill bacteria but not us? Well, penicillin targets enzymes that synthesize the bacteria's cell wall. Now, do animals have a cell wall? Do we produce a cell wall? No. So, because we don't have this enzyme that's key for the function or survival of our cells, penicillin doesn't kill us. Now, there are people who can have an allergic reaction, but it's no different than like a peanut allergy, where the body thinks that it's some toxic substance and accidentally creates an immune response. So for people who are allergic to penicillin, it's not that penicillin is actually dangerous. No more dangerous than a peanut is. But you have these different types of allergens that the body can respond to, and they can be life-threatening for some people, but not because we have an enzyme that this product can bind to. So this is why cyanide kills us but doesn't kill bacteria, because they don't have mitochondria. They don't make ATP this way. So they're like, whatever. But penicillin kills them. That's toxic to them and not toxic to us. So this is where we get into our medicines and our various chemicals that when we treat, that's why we can have such high doses of antibiotics coursing through our bloodstream and not even hurt us, because we don't even have that enzyme that it affects. All right, so I mentioned too that some enzymes are more key than others, but every metabolic reaction in your body pretty much requires an enzyme, whether it's putting proteins together, whether it's breaking glucose down. Every step has different enzymes, and so these are key proteins that are necessary for metabolism. And if you don't have one, it sometimes can just cause mild irritation like lactose intolerance, or it can cause death if you don't have particular enzymes, like with the Tay-Sachs disease. That enzyme doesn't metabolize lipids, it builds up in the brain, and the child dies by about age 3. Now, this second part is more fun because we look at it as application. This second part actually addresses how cells maintain homeostasis by either working with or against entropy. Okay? So remember, When I say with entropy, or we say high to low concentration, that's with entropy. Another thing you're going to see is, in some of the vernacular they use, is what we call concentration gradient. What does that mean? It means that when you have a membrane and you have a high concentration on one side and a low concentration on the other side, that's its concentration gradient, kind of like a hill. It goes from high to low. On one side it's high, the other side is low. If we talk about going low to high, that's what we call against its concentration gradient. And that requires energy. So that's a lot of the terminology I'm going to use. If I say with the concentration gradient, I mean with entropy, with the natural process as it goes from high to low. That's why I use the example of the hill. If I say against the concentration gradient, this is where energy is required, because it's like trying to push a boulder up on the hill. It's trying to go against what naturally occurs. This is where you take molecules and you pressurize them. You put them into a high concentration. It requires energy to do that. So the natural tendency of all molecules in our universe is to undergo a process which we call diffusion. Diffusion is just the inherent movement, which is kinetic energy, within an area to where the molecules try to distribute themselves evenly. So a prime example of this is just making a tea. You put the tea bag in the water, and over time the molecules start diffusing until you have a nice homogeneous solution of the tea chemicals, and that's diffusion. Okay, this is the natural process. It's evaporation of a perfume into a larger area. It's just how everything works. Everything goes from a high concentration to a low concentration. That's diffusion. But when dealing with cells, because they have a membrane, that membrane selectively allows what comes in and out of the cell. So there's about 6 modes of transport that you're going to learn today, of how a cell is able to regulate access to its cytoplasm. Now, some things occur naturally, which is, as we talked about, with entropy. And these are actually key and important processes that you really don't give a second thought to on a day-to-day basis, but they're just constantly happening. So let's talk about the first 3. The first 3 belong to a group of transport which we call passive transport. The reason why we call it passive transport is because all 3 of these require no energy to occur. Why? Because it's the natural order of the universe. All of these occur when you have different gradients of molecules and they go from a high to a low. Okay? So that's why we call it passive transport, is because it occurs passively or without any type of effort, and it's just one of the laws of our universe. All right, so let's talk about the first one. We don't just call it diffusion, because diffusion is just the natural process of molecules to go from a high to a low. When it is across the cell membrane, we call it simple diffusion. So in simple diffusion, the membrane, let me pull up some of my animations that I've created here. The membrane is very porous in the sense that there is space that allows some molecules to go through the phospholipid bilayer. Okay, there we go. So remember, phospholipids are these molecules that have the hydrophilic heads and the hydrophobic tails. And they form this bilayer that encases all of the cytoplasmic components of the cell. It's organelles, it's fluid, and the like. Well, molecules, if they're small enough and they're not polar, can easily just squeeze in between and move through the membrane. The membrane doesn't do anything. In fact, it can't do anything against this. So oxygen is key for our cells to be able to undergo certain or metabolism, or what we call aerobic respiration. Every time you expand your lungs and take in a large concentration of air, what you're doing is you're increasing the oxygen concentration around the cells in your lungs. Well, when the blood reaches the lungs, the oxygen content is very low. And so by having a high concentration of oxygen in your lungs and a low concentration of oxygen in your blood, the oxygen naturally diffuses through the membrane because it's trying to equalize. It's trying to diffuse so that both sides of the membrane have equal amounts of oxygen. But we keep breathing out and reestablishing that high concentration of oxygen in our lungs. So every time we breathe in and out, we keep making it so that more oxygen is on the outside of our blood, and so it diffuses into our blood naturally. So this is what it's illustrating. When you have high concentration of oxygen on one side, it just leaks right through the membrane to try to equalize and become equal. But because we keep breathing out the air and breathing in new oxygen, it'll never equalize. So that's why breathing works the way it does, why oxygen is able to diffuse into our blood through what we call simple diffusion. Well, the same process occurs, but in the opposite direction for carbon dioxide. When we undergo metabolism, the glucose and the sugars and the fats and the proteins that get broken down get turned into carbon dioxide. Well, our cells pick up that, our blood picks up the carbon dioxide and takes it to our lungs. Well, as long as you're in an environment where the carbon dioxide you're breathing in is much lower, then carbon dioxide leaves your blood. And this is the reason why you don't enclose yourself in a sealed room. Because not only does the oxygen run out, but the CO2 starts building up as well. If the CO2 builds up enough, then what ends up happening is you don't get simple diffusion of the carbon dioxide out of your blood. It stays in your blood and it starts causing massive problems. So you need to be in an environment where the carbon dioxide is substantially lower than what's in your blood so that it can naturally diffuse out. There's no other mechanism to do this. Carbon dioxide just will go from high to low. So as the blood reaches your lungs, it essentially diffuses in the other direction. So it all just depends upon the concentration gradient. If oxygen is higher on the outside of the cell than on the inside, it diffuses into the cell. If carbon dioxide is higher on the inside of the cell than on the outside, it diffuses out of the cell. Now, here's another thing you have to be aware of. Every molecule has its own concentration gradient. What I mean by that is, let's say you have your phospholipid bilayer here. You have high levels of oxygen and high levels of carbon dioxide. So carbon dioxide is at high levels inside and oxygen is at high levels outside. They don't look at each other and say, oh, well, because we both are high, then we'll just stay where we're at. Oxygen merely looks at other oxygen in terms of its diffusion. It only deals with other oxygen. And the same thing with carbon dioxide. It only, moves in accordance to its own concentration gradient. So that's why you can have these different gradients and simultaneously oxygen will move in as carbon dioxide is moving out. So you don't have to breathe twice for every type of exchange. You breathe in, oxygen diffuses into your blood, carbon dioxide diffuses out of your blood. You get rid of that air, you breathe in a new batch, same thing occurs. All right, so that's what we call simple diffusion. Does not require any energy on the part of the cell. The membrane is there, and the molecules simply work with entropy to go across the membrane. Now, I mentioned that these molecules must be small and not charged. The reason, because this region right in here, where the phospholipids are at, is extremely hydrophobic, which means if you have any charged particles, ions, polar molecules like glucose and whatnot, then they're actually impermeable to going through this membrane. Let me show you another video. If there is these charged particles, they essentially bounce right off the membrane because they can't squeeze in between. Because of how charged they are, they get trapped on one side of the membrane. So this brings us to our second mode of transport. We call it facilitated diffusion. This requires help. These molecules can't naturally go through this phospholipid bilayer. So they need some type of channel by which they can go through naturally from a high to a low. These proteins are usually what we call ion channels. We also have channels for other molecules like glucose channels and whatnot. So there's a lot of proteins that each are specific for a particular molecule. You have hydrogen ion channels, sodium ion channels, potassium ion channels, glucose channels. So each protein is specifically designed to allow a certain molecule through the membrane. But the key here too is it's like a revolving door in the hotel. The door doesn't move itself, it merely moves when the molecules are putting pressure against it. And so it naturally occurs due to the diffusion of these molecules. So the only difference between facilitated diffusion and simple diffusion is facilitated, the molecules need some help to go through because they're charged or they're large or they're polar and they can't go through that membrane barrier because it's so hydrophobic. Okay? So you might have ions that are much smaller than molecules like oxygen and carbon dioxide, but they'll bounce right off the membrane. They can't go through unless they have some type of pore or channel in the membrane that allow them to go through. That's what this video is illustrating, that they will essentially just travel through the channel from a high to a low concentration. They're at a high concentration on one side, you give them a way out, and they will take that way out because that's the natural order or process called entropy. All right. Now, the last mode of transport is actually the same as simple diffusion, but it's called osmosis. So why do we distinguish between the two? Simple diffusion is the diffusion of any molecule across the cell membrane except water. Now, why do we have to make the distinction between water? Well, it's because simple diffusion is occurring in the watery environment, and osmosis is the simple diffusion of water. Okay? But there's some strange things that kind of go on with osmosis when you look at it. So that's why we kind of separate it in terms of talking about it instead of just simple diffusion. So the same principle applies here with osmosis. If you have high concentrations of water on the outside of a cell and low concentrations of water on the inside of the cell, which way will the water flow? It will flow out or in. It's low here and high outside the cell. It'll flow in, okay? Just like all these other molecules, it'll go from a high to a low. Well, the confusing part is we look at the cells and we look at the solution that they're in, and we don't actually look at the water, we look at how much solutes are in there. For example, let's say you had a 1% salt solution out here. Well, what percentage of the water then is it? If you're looking for 100%, what would the water be? 99%. Okay, so that's the percentage of water, 99%. Let's say inside of the cell, it's at a much higher salt concentration, like 5% salt. How much water do you have? 95% water. So, 99, 95, which way does it go? It goes in. Again, this has less water. So it's the concentration gradient. If the outside solution has 99% water and the inside solution has 95% water, it goes in. What if I reverse this? What if this were 99% and this were 95%? Because the outside was extremely salty and the inside was not so salty. Now which way did the water go? It leaves the cell. Okay, so that brings us to a key aspect of what cells do to try to maintain homeostasis, especially where we're just completely surrounded with these different concentrations of water. We call it tonicity, which is essentially the relative concentration of water out and inside of the cells. Now, if the solution on the outside of the cell is isotonic, that means that the concentration of water is equal on both sides of the membrane. When that happens, the net flow of water is zero. Okay, so iso means the same. So in an isotonic solution, which is what our body tries to maintain in our blood and in other areas, the net flow of water is absolutely zero. Because what happens is, if you have these two, which we call hypotonic and hypertonic, then problems start happening with the cell. What am I talking about? Well, here's an example in your blood. Your blood, if you look at it from the side, looks kind of like a donut without the hole in the middle. It just looks like this. You can kind of see that groove in there. It's not an actual donut shape. Doesn't have a hole in the middle of it, but it is, more round on the outsides and less round on the inside. That's just the shape of it. Okay. Well, when your blood is isotonic with your blood plasma, which is the fluid that it's bathed in your cardiovascular system, then the cell functions normally. But let's say, for example, you lose some blood and you go into the hospital and they put an IV into your blood because they need to restore some of the fluids. The IV must be isotonic with your blood. It cannot be pure water, because if it were pure water, this is what would happen. Your red blood cells would literally blow up like a balloon and burst. So you could kill someone by pumping pure water into their bloodstream. Water kills in many different ways. But this is one of those scenarios, is that an IV actually has salts and sugars in it, so that it's not 100% water. Because the inside of your cells aren't 100% water. There's salts, there's other things in there. And so we measured it, and we know exactly what percentage. It's usually about 2.7% or so of solutes, so that it's 97.3% water or whatnot. And that's isotonic with your blood. So if a nurse were to come in and be like, oh, your IV's out and we're out of stock, well, you still need fluid, so I'm just going to fill your IV up with tap water. You better get out of there because she's going to kill you. Okay? So on the flip side, if you put a solution that has a high concentration of solutes, like salts and sugars and whatnot, and it's too high, meaning it has more solutes and less water, then this happens. Water leaves your cells, the cells shrivel up, and they die, and they don't function properly as well. So that's why if you have pure water put around your cells, or salty water put around your cells, you got a big problem on your hands. Now, plants tend to like being in what we call a hypotonic surrounding. Why? Because they don't burst like our cells do. Why? They have a cell wall. And that cell wall allows the cell to actually engorge itself with water, which is actually critical for its ability to undergo photosynthesis, without dying. The cells don't burst open. So it loves that hypotonic surrounding. If you want to see this at home, most people know this, that if you want to keep lettuce from wilting, then you put it in water. You put it in your fridge, you put it in water. Why? Because when you put it in water, the cells swell up, and it allows them to remain very turgid. However, if you don't give your plant a lot of water, or if you give it water with too high of a solute concentration, then water actually leaves the cells, and you have a very sad-looking plant. So, one of the confusing things that people have with this is when they see the word hyper usually means what? More or less? More. And they think, oh, it has more water in it. No, this is referring to the solute concentration. So a hypertonic solution is one that has a high solute concentration, and as such, less water on the outside of it. A hypotonic solution is one that has a very low concentration of solute, and therefore has a higher concentration of water. This is the one that just people tend to struggle with a lot. Okay? The cell actually does a number of things that work against entropy to try to maintain homeostasis. And so here, these last set of processes actually work against entropy. The first of which is called active transport. So whereas passive transport just happens naturally, active transport is like pushing a boulder up a hill. This requires energy because you're working against what would naturally occur. What am I talking about? Well, the main thing that cells do in certain situations to work properly is they'll actually take molecules from a low concentration and push them against their concentration gradient. So why would the cell ever want to do that? Well, let me give you two examples. The first example is how your neurons work. Your neurons actually have, if you were to put an electrode on the cell membrane of your neurons, it would measure negative 70 millivolts. There is actually a voltage, a membrane potential to these neurons. And the reason for that is because it maintains different gradients of sodium and potassium on each side of the membrane. Well, remember that each one only cares about itself. And so this enzyme here will push proteins against their concentration gradient here for sodium, and it pushes potassium against its concentration gradient the other way. So this one actually has a dual role. It actually shoves sodium out and then shoves potassium in, both of which are against their concentration gradient. So it requires energy to do this process. But by pushing sodium out and potassium in, it creates like a battery, an electrical potential that allows your neurons to do their job. This is why your neurons in your brain use so much ATP. Is in order for them to fire and function the way that they do, it has to constantly maintain this gradient. And it reestablishes this polarity every like 5 milliseconds. Okay, so that's pretty fast. Now, your muscles are another example of how active transport is used in the functioning of your cells. They, when calcium is present in your muscle fibers, your muscles contract. But you need to let your muscles relax as well. So the only way in which you can do that is to remove the calcium. So your muscles actually take the calcium that's surrounding your muscle fibers and push them into these membrane compartments where they're not touching the muscle proteins. That's active transport. So they push it from a low to a high. They concentrate your calcium into these compartments, and then when the muscle gets the signal to contract, then you get facilitated diffusion. It naturally goes from a high to a low. So in one situation, when your muscles contract, you get facilitated diffusion. When your muscles relax, you've got active transport. This one requires energy, like ATP, for that to occur. This one, no ATP is required because it naturally occurs from a high to a low. So that's how your muscles work. Every time your muscle contracts, that's facilitated diffusion of calcium. Every time your muscles relax, that's actually using energy. Now, the last two are actually pretty simple. They're what we call bulk transport. We don't always just pump ions and water and molecules across the membrane. Sometimes you just need to take out the garbage. You just need to have these large mass of molecules that are taken out all at once. So what the cell will do is surround these molecules in a membrane vesicle, and then it fuses these membranes together and releases contents outside of the cell. Again, this is this requires energy for one main reason. When you fuse these membranes, because the hydrophobic region doesn't like the water, there are proteins in here that kind of come together and then they cause the membranes to fuse. That takes a substantial amount of energy to do that. It doesn't occur naturally. The membranes don't just form together like bubbles necessarily do. Okay, so there are proteins here that facilitate this process, but that's where the energy requirement is here. This vesicle must fuse with the membrane. That requires a substantial amount of energy to do so. So exo stands for exiting or leaving the cell. So what's an example of this? Well, when your neurons communicate with one another, if you've ever heard of what we call neurotransmitter release, that's exocytosis. So when a neuron synapses with another neuron, at the end of this neuron, they have these little vesicles that have what we call neurotransmitters in them. Well, when the signal tells this neuron to release that, then these fuse with the membrane, release the neurotransmitter into this area, it binds to this, and then the signal continues on. You know, you've got things like dopamine and serotonin and whatnot. These are neurotransmitters. That's exocytosis. That's how your neurons communicate with one another. This is also used in like lactation, where the breast cells will produce the milk proteins that then get released and go into the lacteal ducts. You actually saw this in your previous lecture on the organelles. Remember the endomembrane system? Right here. This is a prime example where the proteins get made, they get packaged up in the smoothie R, modified in the Golgi apparatus, and then they fuse with the membrane and they're released into the milk ducts. That's exocytosis. So, that's what we call bulk transport. Now, what about bringing things in? Well, that's what's called endocytosis. So, endocytosis is also bulk transport. Prime example of this is one cell eating another. Okay? White blood cells eat bacteria. They even eat cancer cells. Notice that they literally have to form their membrane around a large molecule or a small little series of molecules, but the fact of the matter is it requires a substantial amount of energy to rearrange the cytoskeleton to be able to do this and to form this membrane vesicle. So endocytosis also requires energy to occur for this process to occur. Okay, this is an electron scanning micrograph showing you a lot of the distortion. This is a white blood cell eating a cancer cell to engulf it, and then it will digest it with its lysosomes and then use that material and break it apart. So exocytosis is bulk transport out of the cell. Endocytosis is bulk transport into the cell.